it'll say usually like N or L, within limits, normal, something like that. Now you'll know. You'll be able to say, well, they're telling me my hematocrit. Well, what is my hematocrit? Now you know. Packed cell volume, those red blood cells, the ones that are settling out to the bottom. What's supposed to be my hemoglobin? What's hemoglobin? You're going to know. What should be the mean red blood cell count? What about my platelets? What about my white blood cells? You guys are going to now know. Because if you're in your profession that you're going into, one of the biggest things you're going to be doing is looking at a patient's chart. And for the most part, what have doctors done to learn information about the patient?
Tested.
They have tested. And a lot of that is based on lab work, x-rays. Then you're going to have the family member going, well, can you tell me what's wrong with Aunt Sue? Is your name listed on the HIPAA? Okay, well, let me check. What's your name? Jeanette? Okay. All right, so you're listed on the HIPAA. Now, what is it you'd like to know? Now, I'm going to say, well, I don't understand why her heart is racing, or why she's lethargic, or why she might be spastic, or why because one of the things that happens, all right, when people's electrolytes go out of balance, and I'm not saying this to be funny, I'm saying this because it happens. They can go what people term crazy, okay? Because they talk out of their head, they don't know what's happening to them, they might do stuff that's really weird, and it could be something as simple as an electrolyte being out of balance, especially in older people, okay? Because their bodies are way more sensitive to some of this stuff. Well, they might have cancer. They might have kidney failure, something. Hopefully, you, as the caregiver, are going to be able to look at this and you might see, well, hemoglobin is way down. Iron content is down. That means their oxygen levels are low, which are going to affect every cell in their body. Hopefully you'd be able to look at it and say, well, this is something right here. Have you guys ever heard of this? You're going to need to know this, because this will be on lab reports. An osmol, okay? Because y'all don't have to take chemistry anymore, right? Do you have to take chemistry? Are you taking chemistry now? Have y'all talked about osmoles? Okay, can you tell me what it is? I'm going to make it real simple. So from this point, you're going to know. Okay? An osmol. Does everybody know what that is? You don't know what this is?
Like glucose?
It's glucose. When we eat, whether we take in glucose directly or we take in sugars that have to be broken down into a glucose molecule, okay? Because glucose is the only sugar molecule the body can use. Does this break down in the blood or the fluid of the body? Or does it stay in its form and makes its way to extracellular fluid so that it can go into a cell and be used in cellular respiration to produce ATP? So does it break down or does it stay whole?
Sounds to me like it stays whole.
It stays whole. So an osmole means that that product keeps its complete structure. So if I had 5 glucoses in a liter, which is 1,000 milliliters, I would have 5 milliosmoles. Because if I have 5 of them in that liter, that means they kept their property, didn't break down, and they stayed 5. So this would be 5 molar of the milliosmoles. Does that make sense? Now, let's look at this.
Salt.
Salt. What we tend to think of as salt. We talk about this one because it's one of the easiest ones to learn about. But in reality, salts contain sodiums, potassium, magnesiums, all that sort of stuff, okay? When you take sodium chloride, put it into a water environment, i.e., our bodies, does it remain together or does it break apart? They break apart. So I now have 2 products that stay in the fluid environment. So I would have 1 osmole of sodium and 1 of chlorine.
So for osmolarity, is it the whole of each thing? Will it break down each thing, or will it just count everything as a
In this
whole?
one, break down because each one of these stays whole. So if I only had 1 of each, it would only be 1 milliosmole. But let's say I had 100 of them. I now have 100 milliosmole. Okay? That is something that you're going to learn more about when you get into your programs, because that is going to be a value that will be on the patient's chart. Okay? So now, this is just a smidge of the stuff that we can know about for their numbers in the blood. So that person who is a hematologist, for example, if they're taking a slide with a drop on it, they have to count it.
In 10 views.
Yes. They have to take that slide. They actually have what are called counters, because it will take their view in their microscope, and it will be in squares, and you count per square.
It's gonna be the most boring job.
I gotta take
Boring.
hematology next semester, and we started.
Boring, but important. Very important. It would take somebody with a ton of patience to do it. And I don't mean like a patient in the bed. Patience, CE, okay? Which I definitely have a shortage of. So, it's an extremely important study, for sure, okay? The plasma, which is our matrix, okay? Don't forget that. Plasma is our matrix of blood. A matrix, if you guys remember from part 1, it's usually the area where you find a lot of materials. So the matrix for blood contains water, proteins, which is kind of interesting, nutrients, electrolytes, nitrogenous waste, hormones, and gases. That's everything that should be found in plasma. Water, proteins, nutrients, electrolytes, nitrogenous waste, hormones, and gases. The fluid that gets left behind after the blood clots is called our serum. Ever heard of that? All right, serum is usually referred to in blood tests and so forth. The difference is it's identical to the plasma, but it does not have one of the proteins that should be carried, because the protein that contributes to blood clotting is fibrinogen. That's a protein that contributes to blood clotting. What does it make you think about? Fibrinogen. Fibers. Because the 3 fibers of the body are what? Collagen. Well, elastic. Reticular. Collagen, elastic, reticular. Can I have either of these fibers simply flying around in my blood vessels? No. I don't need little strings moving through the blood vessel. But if I need them, I need to be able to give rise to them. So guess what fibrinogen can do? Give rise to the fibers. If that happens, it creates a clot. Serum is what gets collected a lot of times to test. They can actually run tests on serum of the blood. I will have in my plasma proteins that are very important for clotting of the blood. Under normal conditions, I don't want my blood just clotting anywhere at any time, but I want my blood to clot when it needs to. For example, a paper cut that breaches the inside of my body to the outside world. I want that to clot.
So does baby aspirin mess with the fibers?
Yes. Well, it's going to mess with some of our clotting proteins, thromboxane.